We are starting off with May-June 2017, paper 2-1. List 3 characteristics of bacteria. What would be the answers you would go with? So, bacteria, they are known as prokaryotes, right? Bacteria are known as prokaryotes. Pro means no. No what? No true nucleus. What does this mean? No true nucleus. If you look at the cytoplasm of a bacterial cell, they have a single, they have one chromosome, and it is basically a circular loop, and it is not enclosed within a nuclear membrane, right? So this nuclear membrane, which is there in eukaryotes, that is the cells of plants, animals, fungi, and protocots, this nuclear membrane is not there in prokaryotes, bacterial cells. So we say that the chromosome is basically present free in the cytoplasm. So pro means no. No what? No true nucleus, right? So one of the answers you can write is no nucleus, right? So even your cells are microscopic now. You can say smaller than a plant or animal cell. Right? You can say smaller than an animal or plant cell. That is acceptable, right? Smaller than, that's a good point, but you can't say they are microscopic. Huh? What the hell is going on today? Smaller. So every cell is microscopic, no? So the whole organism is microscopic. No. Smaller than plant or animal cells. So it has a long tail. From where did bacteria grow tails? This fellow and his, I don't know what. Okay, that tail you're referring to is known as a flagellum. Flagellum. Even the sperm cell has a flagellum. Tails? Flagellum is not a tail, fool. It's basically a structure that allows them to. You can put it on silent, no problem. That's okay, don't worry. It's okay, don't shiver, don't worry. I'm not going to eat. I don't eat, even though that I look very fierce and mean. Abdullah? Right. So, no true nucleus, smaller than plant and animal cells. They have a flagellum. You can. There's nothing called a tail, man. It's basically a structure that kind of moves up and down, kind of floats like this, wags like this, in order to allow the bacterium to move. It's not a tail, man. You don't. That's wrong. Not tail, like just right flagellum. If I call your projection of flagellum, is that correct? Okay. Okay, now don't choke now. Right. Okay. So now next, cell wall, but it is made out of peptidoglycan. The cell wall is not made out of cellulose, right? In a bacterium, it's made out of peptido. Hello. I'm out of copies. Yeah. Murine and peptidoglycan, you can use it interchangeably. The textbook or the marking scheme will say murine or peptidoglycan. Both are acceptable. So peptidoglycan and murine, which is basically spelled like this, is basically the same. Made of, sorry. Wait. Shafiq, can you please take, have you not written it and answer it? Can you please get about 3 copies and come on the back? No, of the paper. We're out of copies. Okay, but I didn't ask. That's okay. What to do? And it's wrong. Quickly. Yeah. Actually, make it, I think most students might come. Make it 5 million. Oh, wait, Shafiq, come. Shafiq, come. Give them a call and I'll tell them. I'll consider. It's a time. No, I was not. Rahman, I want about 5 more copies of a past paper. Shall I resend that email? Okay, okay. Thank you. Okay, guys, I'll start separating you. Don't talk, right? So we will be now, we are, drive, I'll be getting your copies. You just write the answer somewhere for now. Later on, you copy come to. Are you Mariam's yes. Brothers? I got 5 copies. 1, 2, 3. 2 people came, 1 there. I ordered extra. Right, the papers are coming. So, right. So until then, you guys write down the answer somewhere. I'll get it to you. Okay. So, cell wall made out of peptidoglycan or murine. Okay. Now, guys, listen. Bacteria have no mitochondria. Okay, good. So basically, yeah. So murine. There are no. You can say there are no organelles. They don't really have. Well, that I don't accept that. To be honest, no. See, this is the thing. You can't say that they have no organelles. No, you can say that they have plasmids. Okay, they have plasmids. No. So why am I listing more than 3? So you guys know the different acceptable answers. So when I write down all these things, you know that bacterial cells have these. Write it down quickly. Huh? They have plasmids. M-U-R-E-I-N. They are single celled. And you can say they have a circular single chromosome. Yeah, some can. Yeah. See, the thing is, not all bacteria can carry out photosynthesis. It's a quality of a few special species. So yeah, better avoid it. Because when you look at when bacteria are generally considered heterotrophs now, because decomposers are generally bacteria. Decomposers are bacteria. Decomposers are either fungi or bacteria. They're cells that basically obtain their nutrition from dead or decaying organisms. That's why they're called saprotrophs. Better to avoid this photosynthesis thing. It's actually not there in the mark scheme either. This photosynthesis thing is specifically for this particular bacteria, this particular species. So it's best if you stick to a feature that is specific to, that is common amongst bacteria. Now, if I say bacteria have no nucleus, is common, right? If I say they are smaller than plant and animal cells, common. It's a fact. They have a flagellum, common. Not every bacteria has a flagellum, but it's a common feature. Cell wall made of peptide or glycan or murin. Basically, all bacteria have that. Have plasmids, single cell, circular single chromosome, all of that is common. So it's best if you stick to those very specific common features. You may be right, but mark scheme doesn't accept it.
Sir.
Yeah.
Sir, only organic source of energy.
Sperm cells, you mean? Yes.
Sir, what is most?
Amongst bacteria, man. And are you high today? Okay.
One is chlorophyll.
Yes, you can go with chlorophyll. Yes. So chlorophyll. Actually, the reason is I'm hesitating before I say these things is because your A-level syllabus says something. Now, what your A-level syllabus actually says is they have a pigment that is not exactly chlorophyll, but like chlorophyll. Understood? And they don't have chloroplasts. They don't have chloroplasts. They don't have mitochondria. They have ribosomes, but of a different size. So for your level, the mark scheme, if you were to say they don't have organelles, they will completely accept it. But if you go, for example, AS, you cannot say they don't have organelles because they do have organelles. Because you're not expected to know that much detail. You're allowed to be inaccurate with your answers, with your knowledge level. So writing no organelles is accepted for your O levels, but I'm not going to do that because I find that personally kind of controversial. So suggest the name of the chemical. So the reason that now you can see spirulina is green in color and is able to synthesize its own food. So what is the pigment that you know of that is green in color and allows what is synthesize its own food? What is the process? Photosynthesis now, right? So the photosynthesis is a process that allows plants to synthesize their own food by combining carbon dioxide and water. You have not talked about any other chemical reaction in your biology syllabus where an organism can make its own food other than photosynthesis. Photosynthesis is the only chemical reaction that we have discussed that enables an organism to make their own food. We didn't talk about anything else, no, right? So the first thing that should come to your mind is photosynthesis. And when they talk about this green pigment, they're basically hinting at you and telling you, okay, what is the green pigment we've talked about? Chlorophyll, right? So chlorophyll is the answer. And we write down the equation for photosynthesis. So carbon dioxide plus water will give you glucose plus oxygen. This is the word equation. Sorry, I've not.
CO2 plus H2O plus.
Yes, very good. Carbon dioxide. So if you're going to, this is the word equation. If you're going to write in symbols, CO2 plus H2O will give you C6H12O6 plus oxygen. To balance this equation, very simple, guys. Where is the key? There's a 6 with the carbon. That means you put a 6 here, a 6 here, and a 6 here. Very simple, right? That's the hint in case you don't remember. Spirulina can be eaten by humans. The United Nations World Health Organization made the following statement about spirulina, right? So it can be eaten by humans. For WHO, spirulina represents an interesting food for multiple reasons. Rich in iron and protein, it can be given to children without any risk. We at the WHO consider it to be a very suitable food. Use your knowledge of animal nutrition to suggest and explain why WHO considers spirulina to be a very suitable food. Superb question. Why is it that this is considered a very suitable food, guys? People online, what do you think?
They are like they're copying everything and waiting.
Okay, so you attempted so.
I failed, but
Okay. Spirulina is a bacteria, man. Vegetable. So what's the reason? People online.
because it contains nutrients, protein, and iron. That is nutrition.
Okay, good. Now use your head and tell me how that is important.
Protein is important.
If you don't take something, it comes like something.
Be serious. Go. Okay. So the key, yeah, go try. Give it a try.
Yeah.
Huh?
Yeah.
No, so this WHO, which is the World Health Organization, which is the branch of the United Nations, right? It has made a statement saying spirulina represents an interesting food. So that means you can eat this bacteria as food. Why? Because it is rich in iron and protein. It can be given to children without any risk. So they are asking you,
That means that
why does WHO consider spirulina to be suitable as a food? What is spirulina contain that make it suitable for you to eat? That means for us to eat spirulina, there should be some advantage. No, not like it's giving, it's not like something that is not going to benefit us. There is something in spirulina that is going to benefit us if we consume it. They're asking you, what are those benefits? What is there in it? Yeah. Okay.
in the production of hemoglobin.
Very good. Okay. Iron helps in the in the production of hemoglobin. Therefore, what does it do? Prevents anemia. Excellent. Right. Good. So you know what are they testing here? Do you know if iron is needed or not? Thank you. So give these two to them and give this to Rayu. Right. So the first thing they're asking you is, do you know if iron is necessary or not in your diet? It is. They're asking you, is protein necessary or not? Let's do a quick revision of the main food groups. We have proteins. We have lipids. Guys, where's your knowledge? Carbohydrates. Carbohydrates. And then, so these are the main biological molecules. When it comes to vitamins, what are the vitamins you are supposed to know? Vitamin C, vitamin D, and vitamin A, right? Vitamin A is, it prevents night blindness. Yeah, that's night blindness. And minerals you need to know about is iron, calcium. That's basically it. No. So iron and calcium is what is in your syllabus. Iron is needed for hemoglobin production. Calcium is needed for strong bones and teeth. Wait, hold on. And taking adequate amounts of calcium, what is it important for? It prevents rickets. Rickets happens in which adults or children? Children. Yes. Okay. Vitamin C prevents scurvy. Vitamin D again prevents rickets because vitamin D and calcium both are needed for rickets. Vitamin A prevents night blindness. This is a quick recap, right? Basically, you can't see in low light conditions. No, man. Right. Proteins are needed for what? Growth and repair. Mainly. Growth, repair, production of enzymes, for production of hormones, because all of these are proteins needed for muscle production. Lipids are a rich store of energy. This is one of many points. I'm just writing some of the main points, right? There are many points when it comes to, I would appreciate if you guys write these notes in some textbook or something so that when you write, you're revising. Otherwise, you just come and stare at this and nothing goes into your empty heads, right? Rich store of energy. And carbohydrates also is a main source of energy. Fibers also important. Yeah, fibers are important. What do fibers prevent? Fibers prevent constipation. Good. So minerals, you have iron, calcium. You guys go and revise what is the rich sources. Now proteins, basically the source of proteins is meat. No, meat, fish, eggs, liver. All of these are rich sources of proteins. Lipids, source of protein is milk, fat, right? Certain fruits like avocados, rich in lipids. Carbohydrates, bread, pasta, sugars, right? That you eat, all important. Vitamin C is present in fresh fruits and vegetables and green leaves. Vitamin C. So you can think of this, all these bitter fruits like lemons, like oranges, like grapefruit, right? Fresh green leaves, all are rich in vitamin C. Vitamin D, once again, present in dairy, egg, milk, liver. Minerals, iron, rich in, once again, meats, red meat, liver. Calcium, rich in milk, in butter, in eggs, rich in calcium. Fiber, to get a rich source of fiber, what is it? Mostly plant products, right? Leaves, vegetables, fruits, all are rich in fiber. The main component is in fiber is actually cellulose. And the reason cellulose acts as fiber is because cellulose cannot be digested by a alimentary canal. So this cellulose acts as bulk, which stimulates peristalsis, that basically prevents the stools from being stuck in your, not stuck, but basically what is constipation? You're not passing stools frequently. So adding cellulose or fiber to your diet adds bulk, which stimulates peristalsis. What is peristalsis? Peristalsis is the wave-like contractions of the walls of the alimentary canal. Because of these wave-like contractions, food passes from our esophagus all the way to our anus. It is not some magic angel or jinn or spirit that is moving the food from our mouth to our anus, right? Okay, it's not gravity. It is the wave-like contractions of the walls of our alimentary canal. What are the two muscles involved? Peristalsis. No, man, esophagus is not a muscle. Circular muscle and longitudinal muscles. Yes, very good. Go home, study whatever areas that I'm teaching you, go home and cover it, right? So that now you're getting a revision about this. Later on also you'll get a revision. Store of energy, store. So quickly write this so that we can go ahead with the answer and proceed. So it's not only about answering the questions in this my past paper class. The reason I'm doing it for longer is so we can get a theory revision at the same time. Do the theory, get a theory revision, and also learn how to write correct answers. Not answers that will give me a stroke or a heart attack and send me to the grave at a very young age. Before I even get married, by the way.
Sir.
8:30 food. You guys don't read the emails and SMSs. All of you go to the front office and complain during the break, saying, I didn't get the email, I didn't get the email. Go and tell me. Ade, at least 1 year check. No, you don't have time to check the email. Why? What are you doing to others? You are looking after your 4 wives and your 18 children and your 24 grandchildren. First of all, before you try to marry 4 wives and have 18 children, try to carry Abdullah with you first. Okay, quickly. You're done, right? So go home, read this part of the chapter, human nutrition. What is this? The part about biological molecules and the balanced diet. Read the tute, everything is there. To show you where in the biological molecules and balanced diet. Here, it's all there in this tute over here. Biological molecules, about carbohydrates, about proteins. That's okay. They're all very small amounts of here. Everything is there. The function, the source, everything is there in the tute. Go and read it. What vitamins you're supposed to study about? Vitamin D, vitamin C, right? Vitamin A, I mentioned it in class. Calcium, iron, everything is there. Right? All of this, go home and read. Okay. So the answer for this question. The answer to this question would be spirulina is rich in iron. Now, Do not write this your grade 8 style answers where you go and say spirulina is rich in iron and it's rich in protein. The being rich in iron will enable more hemoglobin to be made. To be rich in protein means more growth. One shot without repeating, just say spirulina is rich in iron, which allows, which enables more hemoglobin production and prevents anemia. Don't say spirulina is rich in iron and protein, then again say iron is needed for this. No, save your time, use as little words as possible, spend as little time making your answers long. You have only, oh yeah, I forgot to tell you at the start of the class, this paper, I can't remember now the total. Is it 80? This is usually how I start the past paper class. Right. So your paper, you have 3 papers that you're going to be doing. You have your multiple choice paper, which will be mostly doing during the month of May, just before your exam. We'll be doing about 4 or 5 papers a day and completing all of the papers needed. You have your theory paper, which is what we're doing here, 1 hour 45 minutes, 80 marks. 1 hour 45 minutes is how many minutes in total? 60 plus 45, 105, right? 100, you have 80, for 80 marks, you have 105 minutes. That means for 1 minute, for 1 mark, how many minutes do you have? About 1.3 minutes. That is about 1 minute and 20 seconds. So for every mark in this paper, you can maximum spend 1 minute and 20 seconds. Right? Do not spend less. No, I mean, do not spend more than this. If you spend more than this, you're eating up the time available, that eating up the time that you should be spending for another question. If you spend on a 1 mark answer, if you spend 3 minutes, that means you're losing 2 minutes that should have been given to another question. So when you do these past papers at home, solve it in a room, 1 hour 45 minutes straight, put a timer, start it, end it correctly on time, and see how your speed is progressing. This is something I cannot do for you. Speed is something you, because over here I cannot solve this paper in 1 hour 45 minutes, right? Because I have to explain things to you. Speed is something you have to develop with practice. So every time you do a past paper, lock yourself up in a room, switch your phone off, tell your girlfriends and boyfriends not to disturb you, right? And if they are, tell me, and I will go and tell your parents, right? And you go and do whatever you have to do in that 1 hour 45 minutes to make sure you solve this paper in the best possible way. At the end of the 1 hour 45 minutes, if you want, you can go through the relevant areas. For example, the question about nutrition, look at the nutrition tune. Question about photosynthesis, look at the photosynthesis tune, and you see where you could possibly be wrong. When you come for my class, you write down every answer that I write. Make sure you write it down, because the goal of the past paper class is not to find. You guys only think, no, how many marks I got? No, it's about how you can write correct answers with the least number of words, so that you're not spent. What you can write in 2 sentences, most of you write in 5 or 6 sentences. That means you're writing 3, 4, 5 sentences unnecessarily. You're wasting your time. Don't let that happen. Keep your answers short and sweet. They don't care about this. You're not supposed to fill in all the spaces. If you can get 3 marks by just writing 1 line, when they have given you 10 lines, they will give you those 3 marks. Make sure you write the points short and sweet, right? So this is how you have to approach your past paper class. You have 1 minute, 20 seconds for every mark. Be very careful. We have an alternative to practical paper, which is paper 4, right? Which is for 40 marks. It's for 1 hour. It's not a very difficult paper. It is the theory paper that is considered usually difficult. So make sure this is your approach. I'm going to be giving all of you the papers for 2 days. For the next 4 papers, I'll be having, I have it with me, right? The next 3 papers, sorry, after this paper, the next 3 papers have all been printed out. It's going to be given to you. Make sure, don't wait till I tell you. Let's say we have finished today's paper, this paper today. Don't wait till I tell you to do the next paper. Do it. I'll tell you which order it should be done. I'll put a message on our group today. Make sure you follow it, right? So once we get now, after this paper, now the paper, how do you know what paper you're doing? If you look at the top top right of the paper, it will be written May, June 2017, right? Paper 21, 509021 over here. 509021, right? So the next paper I'm going to be doing is, everyone keep it in mind, paper 2 May, June. Then I will be going to October, November, and I'll be doing 2-1, and then I'll be doing 2-2. It will be covered in this order. All the papers have been printed here. I'm making the effort to print it and give it to you so that you can solve it and come for your benefit, right? Otherwise, I can give you this. I don't have to give you this. I can give you this on the day of the past paper class, right? I'm giving this to you so that you have the paper, so you don't have to waste your own bloody printout, your money to take printouts. So you can get this and you can be prepared and come at your convenience, right? I am thinking, when you first started O-level 2023, I told you that when we do past papers, it will be intense. This is my approach. Why? You can go from an E to an A star if you do your past paper classes properly. You can, even if you know your theory at an A star level, if you don't attend your past paper classes, don't follow what is being taught, you can easily drop to a D, right? So your past paper classes make or break you. This is, and in the last moments before your exam, I don't recommend looking at your tutes. Reading tutes at that point is useless. Read your past paper answers. Because in your past paper answers, not only do we learn how to write the answer, but how to write the answer in a short and sweet way with all the information. Understood? So when you're before the exam, the day before exam, let's say you're having paper 2 tomorrow, you take all the paper 2s and one by one, you look at your answers. Look at the questions and you see how you've written the answers. What answers are relevant? Understood? Right. Having said that, let's proceed. So spirulina is rich in iron, which is essential for hemoglobin production. 1 mark. This prevents anemia. So that is your second mark. Now you've got 1 mark for the first sentence, second for that, right? Protein is essential for growth and repair. As well as, you can say, enzyme or antibody production, as well as enzyme. Don't write hormones. Actually, no, you can write enzymes just, and yeah, enzyme, and you can write antibody production / antibody. When I put a slash like that, it means I'm giving you, saying that you can use either of these 2 words, antibody production. So that will give you 4 marks. We can go on ahead. Yeah. Protein is essential for growth and repair is the 3rd mark. 4th mark is as well as enzyme and antibody production. So you can also say that since spirulina photosynthesizes, photosynthesis produces glucose. Glucose can be used to can be used to store, form basically starch or whatever, right? Spirulina, since photosynthesizes, it will be rich in carbohydrates. So all the green lettering is enough for 4 marks. I'm telling you what are the possible answers. The red part is the other points you can give. And provides components of a balanced diet. So what is a balanced diet? A diet that includes all food groups in the correct proportions. All food groups means carbohydrates, lipids, proteins, vitamins, minerals, and fiber. All of these food groups in the correct proportions. That is, protein, you're taking in protein in the correct amount for your age, for your sex. for your activity. You're taking in carbohydrates in the correct amount, not taking excess carbohydrates, little proteins, little vitamins. That's how you become obese. That's how you get diseases like diabetes and heart disease, right? Okay. Quickly, guys, write it so we can progress. No, it's the extra points.
How many points are there then?
6 in total. So this one will get you a 5th point. This will get a 6th point. Rich in carbohydrates is 1. 6 is components of a balanced diet. But it's a 4-mark answer. I'm just writing excess to show you what else you can include. Right? Okay. Next, a poto. Yeah. Never. Otherwise, there will be people who will not leave even a single inch of the paper empty. They will keep writing.
I put 1, 2, 3 and then.
No need to write. No need to put 1, 2, 3. City has gone off. Okay, guys, concentrate, focus. Right. Okay. Now the screen is okay now? Okay, guys, attention, stop talking. Okay, a potometer is used to measure water uptake by a plant. Figure 2.1 shows the stem and the flower of a plant in a potometer. As water is taken up, the bubble moves in the direction shown. So a potometer basically, in other words, measures the rate of transpiration. What is transpiration? Basically the rate at which the water is lost from the plant, right? Through evaporation. Okay, so you can see a water reservoir, which is basically the water that is, yeah. So anyway, now name the parts A, B, C, and D. What is A? Okay, so we'll just go through the since you guys don't know. Yeah. No, the specific parts with.
So A
Ah. So reproduction in plants, right? Okay. So when you look at the structure, really nice, no? Yeah, very weird that you said nice, because if you said human reproduction, you said nice, no problem. You said flowers, poor flowers are in your gardens. I don't know what you're doing with them, right? Okay, so in your flower now, when there is so when you take a flower, you have these large colorful structures known as petals, and you have these structures right here at the bottom that are sepals. Basically, the sepals, before the flower kind of blooms, the sepals kind of protect the flower, right? Kind of protect the flower. You have the receptacle, which is below right there. And then you have the male and the female. You have the male and the female sex organs of the flower, right? So the flower usually contains male sex organs and female sex organs. So this bit right here, which is known as the stamen, which is known as the male sex organ, right? You have the anther, which is where the pollen grain is contained, basically, and we can say produced, and the filament, which basically suspends the anther in the air, right? Then you have the stigma, the style, and the ovary, which contains the egg cell. Inside you have, so inside the ovary, do I have a picture of that? So inside the ovary, if you have a look at the ovary, you have a chamber or an area known as an ovule, which is where the ovum or the egg cell is located, right? So the male sex organ is known as the stamen, which contains the anther and the filament. The female sex organ is known as the carpel, which contains the stigma, the style, and the ovary. The ovary contains the ovule, which contains the egg cell. So pollen grains, during a process known as pollination, pollen grains are generally transferred from the stamen, or you can say the anther, to the stigma. This could happen within the same flower, that is from the stamen of one flower to the stigma of the same flower. This is known as self-pollination, right? Self-pollination is basically when either the pollen is transferred from the anther of one flower to the stigma of the same one, or the anther of one flower on one tree to the stigma of another flower on the same tree. That is also called self-pollination. Understood, right? Cross-pollination is when flowers of different trees are pollinated, where the pollen from a flower on one tree goes to the stigma present on the flower of another tree. That is cross-pollination. Yeah. So what, you tell me that answer. Why? What is the advantage of cross-pollination compared to self-pollination?
variation.
Huh? What? Good. Yeah. Genetic variation. Why? If you were to reproduce with yourself and produce a child, if you were to reproduce with yourself and produce a child, won't your child be identical to you? They don't have deficiencies. Now you have to talk about your deficiencies also. Okay. So now if you were to produce a clone of yourself, it will have the exact same genes, the exact same alleles. There will be no variation now. What did I tell you when I taught you variation? The more the different combinations of alleles, the more chance of survival. If you remember that example I gave? You remember that example I gave, no? Of giraffes with long necks and short necks when I taught you? If there was a situation, anyway, we'll talk about that variation later. I'm just eating up this time. Right, yeah. Good. You can think that. But also try to be more perfect. Try to be more perfect. Okay. So, guys, right. So, A is a petal. B is a sepal. C is a leaf. And D is the vein. So you take the leaf like this. You have a structure called the midrib in the center from which the veins are located. So the veins basically contain, you can say vein, you can say vascular tissue, and you can say xylem and phloem, right? Well, xylem, don't say xylem or phloem, xylem and phloem. Xylem and phloem. Because both are present in the vein. Vascular bundle is also, vascular tissue is better. Vascular bundle, you can say, yeah, vascular bundle. It's not given like that in the mark scheme, but okay. This, okay. What is transpiration? Wait, I have to use the diagram to explain to you. Otherwise, you won't understand. Okay. So this is your leaf here. Now, in your leaf, you have mesophyll cells, the spongy mesophyll cells particularly. So the surface of the spongy mesophyll cells is basically wet. It is moist, right? Now, what happens is, under certain conditions, this moisture will evaporate the water.
Basically, transpiration is when the what temperature goes up. Basically.
Yes. No, it's not exactly that answer, because if you fools, listen. When it comes to transpiration, you have to be clear, because if you get a 3-mark answer, that you only get half a mark or 1 mark, because that's a pathetic definition.
I'm not disagreeing that way.
Okay. Yeah. Okay, yeah, no. That's the transpiration stream. This stupid projector, I don't know how many times it has gone off. Hmm? Joker. Not exactly correct. Okay, pay attention. What is transpiration? Basically, the surface of the mesophyll cells is basically has a layer of water. Now, what happens is, this water, right? Transpiration is the evaporation of the water on the surface of the mesophyll cells into the air spaces, and the diffusion of this water into the external environment. 2 parts. It's evaporation, that is the water, liquid water is evaporating as water vapor into the air spaces, and it is then diffusing out. Why am I not saying that it is moving out through osmosis? What's the reason? Why didn't I say that it moves out through osmosis? Why? Okay, good. That's one. Very good. Okay, what else? More important thing. Water vapor is a gas. Yeah. Yes. Gas. So water vapor is a gas. Gases move through diffusion. Gases don't move through active transport. Gases don't move through osmosis. Osmosis, the movement of substances through osmosis, the only substance is water. When it comes to gases, it's diffusion. That's why when we say carbon dioxide, when it enters and leaves the blood or whatever, the tissues, it's diffusing. It's moving through diffusion. Water we're talking about is liquid water, darling. This is gas. That's why this is called diffusion. Evaporation of water from the surface of mesophyll cells into the air spaces, and the diffusion of this water vapor from the air spaces through the stomata into the atmosphere. That is exactly what is happening, right? So evaporation into the air spaces, then diffusion through the stomata into the atmosphere. Is that clear? Right. Next. Right. So describe the pathway taken by water as it moves from the potometer through the plant stem and into the surrounding air. So as it moves through the potometer, now there is no root here, no, so you don't have to talk about the root. Where as it enters the stem, what part of the stem does it enter? What is the vessel? So you know that in the stem you have two vessels, xylem and phloem. Xylem carries minerals and water, right? Phloem carries amino acids and sucrose. Xylem carries water and mineral ions only in one direction, that is from the base of the plant to the top. Phloem can carry it in any direction, either up or down or up. It's up, it depends, right? So what you have to write is the water passes, water passes through the xylem to the leaves, to the leaves. Here it passes between leaf cells to the surface of the mesophyll cells, from which it enters the air spaces. Air spaces are located in which layer? Of the spongy mesophyll, right? Then exits the leaf through the stomata.
So what is here? It is passes, right?
Hmm?
The second line here is passes.
Here it passes between leaf cells, the surface of the mesophyll cells, from which it enters the air spaces of the spongy mesophyll, then exits the leaf through the stomata. So to exactly describe this pathway, this is how it goes. Can you see this here, this diagram? Full-time people, I have not done transport in plants, so don't worry. So it comes to the leaf through the xylem, which is right here. Okay. From the xylem, it passes through between the leaf cells. Can you see this? Passes between the leaf cells to the surface of the mesophyll cells. So in the leaf, you have two layers. You have the palisade mesophyll, the spongy mesophyll. The spongy mesophyll has air spaces between them and the cells are sort of round in shape. The palisade mesophyll Cells are somewhat rectangular in shape and tightly packed towards the top of the leaf. So the pathway taken by water comes out of the xylem, moves between the leaf cells, and between the leaf cells, it comes to the surface of the mesophyll cell. From here, it evaporates into the air spaces. Then water vapor diffuses out through the stomata into the atmosphere. This is the pathway taken, right? Can see this here, right? Here, very clearly mentioned, passes between cells. It doesn't really enter the cytoplasm. It's passing between the cells. So remember this diagram, very important, passing between the cells and then surface of the mesophyll cell, then it evaporates, then exits. So all of these answers, everything to your past paper questions, all the answers are there in your tute. Make sure you follow it, right? Okay. So back again. That is your answer. Let's move on. Go and read the tute. Every question that we do, go and read the tute again and revise what was done. Go and read the tute again. The reason I'm going back to the tute to show you what was written is so that you guys get used to reading the tute, because you guys are useless now. You are like buffaloes. Once you finish the tute, you go and put the tute under your bed and you sleep. When close to the exam only, you're looking for the tute to see it's under the bed. The bed, I don't know, cockroaches have mated, reproduced, bacteria have, their great grandchildren are present on the tute, right? Under the bed. So that's why. Huh? Yeah, I was there. Right. Okay. Next question. No. Quickly. Next. In an experiment, the bubble moved a distance of 60 millimeters in 10 minutes. Calculate the average rate at which the bubble moved per millimeter per minute. So 60 millimeters, it moved in 10 minutes. So per minute, how many millimeters? 6 millimeters per minute. Do not forget to write the units. If the units are given, well and good. If the units are not given, they usually give 1 mark for the units, right? Sometimes they will say, if the answer is not given with the units, you get 0. So don't get 0. The experiment was repeated in an area of lower light intensity. Predict and explain what would happen to the rate at which the bubble moved. So now what will happen? This experiment, the experiment they showed was at a high light intensity. Now they're repeating the experiment in a dim area or in a time where there is low light intensity. What is going to be the effect? So this question is about transpiration now. So you have to link two things. What is the effect of light intensity on transpiration? When light intensity increases, what happens to transpiration? Why does it increase? When there is more light, there is more heat. When there is more heat, there is more evaporation. Right? When the temperature is high, transpiration is higher now because of this evaporation happening. Right? When the temperature is low, less heat available, or when the light intensity is low, less heat, so less evaporation of the water from the surface of the mesopills into the air spaces, so less will be lost through the stomata into the atmosphere. So as the light intensity increases, temperature increases because there is more heat provided, right? In the 12 hours in the afternoon and 6 p.m. just before sunset, there is a difference in temperature now. Why? Because of the sun, the intensity of the light. Okay, so the rate reduces and also basically one of the main thing actually which I didn't really mention, the other thing is during bright sunlight, what happens? That allows transpiration. So I forgot to mention transpiration, the loss of water vapor will not happen unless the stomata open up, right? So when will the stomata open up? When the light intensity is high. So it's actually when there is a high light intensity, stomata open up and more water is lost through transpiration. But when there's a low light intensity, the stomata close in order to protect the plant against water loss. So this is actually the main answer, right? Okay, so
Sir.
yeah, tell me. Huh? Mutations can lead to characteristics that allow the organism to adapt to the environment. Adaptations.
It's like stomata only opens up.
So there are certain chemical reactions that you study in your A2, right? Hormones, involvement of hormones as well. Okay, so the explanation is in low levels of sunlight, the stomata will close. As less photosynthesis is taking place. Lack of, because what did we study? Light intensity is directly is a limiting factor when it comes to photosynthesis. Photosynthesis, what did we study? The factors photosynthesis depends on sunlight. Then what? Carbon dioxide concentration. In your tooth, there are many graphs. Third one is what? Photosynthesis. Sunlight or sunlight intensity. Okay, same thing. What else? Last one, you temperature. When the huh? No, photosynthesis. Depend factors that photosynthesis depends on. The fourth one, they say not very important. It should be like, at the bottom of your list, water and chloroplast concentration. Let's ignore this chloroplast con too much for you. Water. We are talking about the factors that affect the rate of photosynthesis. If you increase light intensity, photosynthesis increases because light intensity, the light energy is used to combine carbon dioxide and water to produce glucose. Number two, carbon dioxide concentration. Carbon dioxide is a raw material, is a substrate for photosynthesis, right? That's why carbon dioxide is considered a limiting factor. Less carbon dioxide means less substrates available to make glucose. No carbon dioxide means no glucose, right? Less light intensity means less energy available to combine carbon dioxide and water. Temperature. Why is temperature important? Because every bloody chemical reaction that takes place in a living organism, every biological reaction is carried out by enzymes. You increase the temperature, and if the temperature is close to the optimum temperature, these reactions will happen at the optimum level, the highest rate. That's why temperature is important. Temperature is low means photosynthesis, the rate of photosynthesis will be low. Is the tute to show you, since you buffaloes don't remember. Here. These graphs are there in your tute. Hey, do you guys even read this tute? Light intensity. It's written, I have written it out over here. Temperature, light intensity, carbon dioxide concentrations. I have said, also mentioned, see, depends. Water can be considered a limiting factor. Sometimes it's not considered. The reason why water is not really considered a limiting factor is because if the plant absorbs 1 liter of water from the soil every day, out of this 1 liter, 900 ml is lost as transpiration. So water usually is available in excess to the plant, to the point where the plant is losing more water than it actually uses for photosynthesis. But I've told you when I was teaching you this lesson, if you're writing a list, mention these three first, then you can write water, and then you can write number of chloroplasts. Number of chloroplasts will be last in your list. The three most important factors are those. Carbon dioxide concentration. What are you doing? Carbon dioxide concentration, light intensity, and temperature. Right? So go and read these curves, the effects, what is a limiting factor, all of that. Make sure you cover it. Right? So low levels of sunlight, the stomata will close as less photosynthesis is taking place. As a result, stomata close, and therefore less transpiration takes place.
So not repeating it, yeah. If it is low level of sunlight, it's not a bit closer.
Ah, sorry.
Close, close.
Sorry. As a result, less loss of water through transpiration. In low levels of sunlight, the stomata will close as less photosynthesis is taking place. As a result, less loss of water through transpiration occurs. Right. Next question. Organ X, okay, full-time batch. This is for you. We have already covered this. Figure 3.1 shows organ X and its associated blood vessels P, Q, and R. Organ X is involved in the following processes: the metabolism of amino acids, the breakdown of chemical substances, including alcohol. What is this substance? What is this organ X? Liver. So the role of the liver, one of the roles is deamination. Deamination. Do not say deamination of amino acids. Deamination, detoxification. It has a role in glucose. Glucose blood glucose level, blood glucose homeostasis. That is, it controls blood glucose levels. How? It contains the glycogen stores. When the blood glucose levels are low, glycogen in the liver is broken down to release glucose into the blood. When the blood glucose levels are high, insulin is released, which converts glucose into glycogen and stored in the liver. Homeostasis, detoxification, deamination. You can write here, responsible for production of plasma proteins. We studied about prothrombin and thrombin, no? In clotting, prothrombin converted to thrombin, fibrinogen to fibrin. I can't anyway. Plasma proteins, right? That responsible for production of plasma proteins. So organ X is the liver. What is blood vessel P? That is taking blood away from the liver towards the heart. What is blood vessel P? Hepatic vein, fool. Pulmonary vein is the lung. Hepatic vein. So remember, when you're describing the blood vessels of the liver, always write hepatic, because hepatic means liver. So there are 3 blood vessels you're going to be talking about. The hepatic artery, which is the blood vessel that takes blood from the aorta directly to the liver. That is R, hepatic artery. The second blood vessel you have to think about is the hepatic portal vein, which takes blood from the alimentary canal, especially the intestine. Alimentary canal is what? Digestive system, to the liver. So Q is the hepatic portal vein, and P is the hepatic vein. So hepatic vein is P, hepatic portal vein, and this is the hepatic artery. Huh? Yeah, it's not in your syllabus, so don't. Compare the structure of the blood vessels P and R. So blood vessels P and R. You're comparing, they ask you to compare a vein and an artery. Good. Yeah. So now basically say P has a wider lumen than R. P has thinner walls. P has less muscle and elastic tissue both in the walls. P has valves, whereas R does not have valves. Okay, these are differences between artery and vein. Arteries have a narrow lumen, veins have a wide lumen. Arteries have thick muscular walls, veins have thin walls with less muscle and elastic tissue. Arteries don't have valves, veins have valves, right? Yeah. Huh? It's cold.
Those are things that we-
So, let him suffer, let you adapt.
Adaptation. Adaptation.
He's just talking random nonsense. How many questions?
That's,
How many questions are 4, 5, 6, 7,
Paper day after.
8, 9. So many questions. Okay, next question. Estrogen. Estrogen is an example of a type of chemical substance produced by a gland that alters the activity of one or more specific target organs before being destroyed by organ X. So basically estrogen is produced by a gland. What is the gland estrogen is produced by? In the boys in this class, estrogen is produced by their testicles, right? Yeah. Right? Anyway, in normal people, estrogen is produced by a gland known as the ovary that alters the activity of one or more specific target organs. So, like any hormone, estrogen targets a target cell or a target organ. I taught you this, right, guys? What is a target organ or target cell? Each hormone has an effect on specific cells, right? Now, for example, when it comes to insulin, insulin goes and has an effect on liver cells and muscle cells. What does it tell the liver and muscle cells? Start to absorb more glucose from the blood, so the blood sugar can reduce. Start to convert this glucose into glycogen. Start to use this glucose up through aerobic respiration in order to reduce the blood sugar levels. Same when it comes to estrogen. Estrogen goes and it basically stimulates the development of secondary sexual characteristics. It stimulates and it controls the menstrual cycle by acting on various parts of the reproductive system. So what type of substance is estrogen? A hormone. With a reference to estrogen, complete the table. It is produced in the ovaries. Where else does estrogen act? It can act on the uterus, right? Why uterus? Because during the menstrual cycle, estrogen triggers the repair of the endometrial lining after menstruation takes place. Do you remember that? Do you remember that?
I don't know.
You remember the menstrual cycle? You're the expert here. Yeah. Do you remember this graph? So can you see here? After menstruation takes place, where the lining of the endothelial, the endometrial lining or the lining of the uterus, the endometrium. You can use two terms to talk about the lining of, to refer to the lining of the uterus. You can use two terms to refer to the lining of the uterus. You can call it the endometrium, or you can simply refer to it as the lining of the uterus. During the first 5 days, that is during menstruation, the lining kind of breaks down and is shed. Thereafter, the lining is repaired. It becomes thicker again because of the action of estrogen. You can see estrogen is rising in the graph. When it rises, it causes the lining of the uterus to thicken. Understood? Right. I made you guys draw tables, no, about this estrogen and progesterone and everything. Did you study those tables? Huh?
No, sir.
Study it full. Next time, if you come and without studying, you wait and see. You studied it. You forgot.
See what we can do.
Effect on target organ. What is the effect it has on the uterus? It repairs the lining.
Repairs the endometrial lining.
Who has written that note that I gave of this estrogen, progesterone, FSH, LH? Give it to me so I can take a photo and go after this.
Yes.
Good, read this up. This is important. Okay, next. Figure 4.1 shows a Bengal tiger. Fur color in the Bengal tiger is controlled by a single gene. The dominant allele of the gene results in orange fur. A single change in this gene produces a recessive allele, which results in white fur in tigers with homozygous recessive genotype. Explain what is a gene.
A gene is a protein. No. Protein.
Gene is a section.
Words for protein, specific characteristics.
From reproduction even to inheritance. Why? After reproduction is inheritance now. The section of DNA that codes for so codes for the production of one protein, right? So remember, one gene, one protein. That codes for the production of one protein and is the unit of inheritance. Meaning that when you're passing your characteristics from yourself to your offspring, it is the gene that is the one that is being passed on. That is how characteristics go. That's how you get your mother's eyes, your father's eyes, mother, whatever. All of that is through the passage of genes, which is the unit of inheritance. State the term used to describe a change in a gene. Mutation. We talked about two types of mutations, no? Gene mutation, not allele mutation, when that's gene. And we talked about chromosome mutation. Chromosomal mutation. In a gene mutation, a single gene is changed. In a chromosomal mutation, many genes are changed. For example, what is an example of chromosomal mutation I gave you? Down syndrome. Excellent. Down syndrome. And the example of gene mutation? Sickle cell anemia. Excellent. You're reading the tune. Very good. Okay. Right. That is called spina bifida. Yeah, Down syndrome. Okay, use letters. Okay, now all of you do this now. I have taught you this now. All of you do this. Then I will give you a break at 7 for 20 minutes. Hey. Sit and do. You get another one. Wait, be patient.
Sir.
Now, I taught you a particular way to draw this monohybrid process. Yeah, no, not only that. Phenotype of parents, genotype of parents, gametes, everything I taught you, do it like that. Anyone who doesn't do it properly will get a nice shot on the head. Do it. Cool. Do it. How I taught you? Yes. I should not be telling you hints. You're supposed to know this.
So basically, the experiment of acupuncture use the same
They're asking white fur. How can orange parents produce white fur tigers? Yeah. So that is up to you to decide. I'm not going to tell. Practice. What did I tell you? An allele is recessive if the parents don't have now. If the parents are having orange fur and the children have the white fur, it means that white fur is usually recessive. I told you how that happens. How can a characteristic not be that if there is not the parents don't have white fur, how can the children have white fur? Two ways, no. Some other person is the father, right? That's the way I told you, no. Or in that answer is not valid in biology. Then the correct answer is what? How can the child have the features that the parents don't have? That means actually the mother or father is different, no?
Father is heterozygous.
No food, yeah, okay. That's
And heterozygous gene.
yeah, you're right. Gene? Genotype fool. Genotypes are heterozygous and homozygous. So now you gave me answers now during the class. No, sir, father is different, not the real father, right? That is not accepted here. You have to talk in terms of, you are the one only who said that. Yeah, I'll give you one. You not right, do stop talking and start here. How I taught it to you, I made you fools draw the Punnett squares, and I told you based on the combinations, homozygous or heterozygous or whatever, to remember the ratios. Yes or no?
Yes, sir.
Yeah, you say yes sir, but you don't remember anything. Study and come for the next class. Why the answer is there? Go. Come here. You get 0 for this. This is not how I taught this to you. I taught you a nice order, no? Throw it.
I don't get it.
Okay, the combination is correct, but nicely I told you, no. Come here.
I told you.
I won't do anything. Come.
How you did my head.
Also write it. You will not even get 1 mark for this. Go erase it and do it again.
Sir, with the specified thing, you put acute can clear also.
That means you also got the same answer. Automatically you also. Come show me then. Show me the answer.
Can you please, or it can be like.
Normal, they ask, this is what I. This is I is for blood group, man.
So they have given T and T.
Do it again. 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 Do it again.
No, it's like, right, because this one will get 5 marks. I don't know, 7 times. Teaching, sir. Discussion. Allow them.
It's not. Who is very
Wait, my husband is what are the
I thought only the full-time batch gives me strokes. To see you, your batch is also giving me strokes now.
I'm sure, but I think
Wrong.
It's not complete. All day you get to nice and neat, but it's correct. So,
Write it properly. This will not even give 1 mark.
yes, sir. What makes nice and neat?
Okay, okay. Now, like lovers don't argue.
Not the down part.
Still not happy because I taught you in a better way. So you are supposed to follow what I taught you in class. You guys are, I don't know what bullshit techniques you're using. Not using the techniques I taught you. Sure.
So it's not.
Correct. Correct. But is it by fluke that you got this? Explain why. How is it possible?
I don't understand the table. I did this by myself. I couldn't do the table.
Why not?
This is wrong. I have done this also in class how many times? Everything is correct. One thing is wrong. There is no phenotype called orange white man. It's not an ice cream. That Cargill's, it's not an ice cream that Cargill's is selling. Half vanilla, half chocolate, half strawberry, half mango. Huh? Use this thing. This one is better, but basics is right. Okay, okay. You take. The correct answer is, first of all, usually they will, sometimes they will not even give you the, sometimes they might not even, sometimes they might not even give you the key. They will not even tell you which allele is for what, right? Because they've already given you a hint here. It says here, a recessive allele is basically the white fur is represented by a recessive allele. So you have to know, okay, capital T means this is for orange, and orange is the dominant allele. Why? Because it's represented by capital T. Simple T represents the recessive allele, which represents the recessive characteristic for white fur. Phenotype, genotype. Parent 1, parent 2. Phenotype of parent 1 is orange. Phenotype of parent 2 is also orange according to the question. Now, if one of the parents was homozygous dominant, will is there a chance that any of the offspring can be homozygous recessive? Yeah. And I'll tell you what that means. So the correct answer, first of all, is this. Two parents, both the parents are heterozygous, meaning that they have the dominant allele and the recessive allele. Because they have the dominant allele is orange, they will have orange fur. But how can they have children with white fur? If the gamete from this parent carries the recessive allele and the gamete from this parent also carries the recessive allele and they end up fertilizing each other, you can have a chance. So let's draw the Punnett square that shows. So next one is gametes. How do you draw the gametes? Like that. This is how you draw the gametes. Then you draw the Punnett square.
Sir,
Yeah.
I will be homozygous.
Sorry? No, never. So let's say now one parent always goes this way. I always tell you, do not confuse. One parent will go this way. So I'll put parent 1 like this. Parent 2 will go like this. Then I start filling in the box. When you're drawing the gene, so this combination of alleles is known as the genotype, right? So this, wait, homozygous. What is this genotype called? Homozygous dominant. It's called homozygous because same copy, same allele, 2 copies are there. Homozygous dominant. Why is it dominant? Because 2 capital T's. This is homozygous recessive. This is heterozygous. 2 different alleles. Now, what do you see here? You have 3 individuals. The individuals that will have brown fur, I'll highlight. The orange fur, I'll highlight in yellow. I'll highlight in yellow. This will be orange. This will be orange. This will be orange. So 3:1 will be orange. So orange is to.
Both simple things.
Sorry. Yes, sir, sorry. Orange 3 and white 1. Now, this is the only way in which a parent who is showing one characteristic will have a child that is showing a different characteristic, right? Now, why is this not possible? Now let's say one parent is now to show the orange fur, to have orange fur, your genotype can either be homozygous dominant, an individual who has this genotype will have orange fur, and an individual that has this genotype will also have orange fur, no? Yes or no?
Yes.
Individual that has this genotype will have white fur, because white is recessive. And if a recessive characteristic is to be shown, then 2 copies of the recessive allele should be there, homozygous recessive. Right? Now, if 2 parents were crossed, one parent had this genotype, the other parent had this genotype, all the offspring will have orange fur. Why? If you draw the Punnett square, capital T, simple T, capital T, capital T, what do you realize? So these two guys will also have orange, and these people will also have orange, meaning that the chance of all 4 individuals will have orange fur, meaning that there is a 0% chance that any of the offspring will have white fur. Is that clear, guys? Janu, all of you understood? So now remember, this is a shortcut. If they give you a question where the offspring is showing a characteristic that the parents don't have, then 100% the offspring. Now in this question it says 2 orange parents are producing an offspring with white fur. How is this possible? Only way that is possible. What should it tell you? What did I teach you when I was teaching you this lesson? if the offspring is showing a characteristic. Now in a gene, let's say both two parents are having black eyes, the son is having blue eyes. How is that possible? Other than the parents are different, right? The only way is possible is, now this is what is the concept here? The offspring is showing a characteristic that the parents don't have. That should immediately tell you the characteristic that is being shown, the white fur, the blue eyes. It is recessive. Recessive. Offspring is showing characteristics that the parents don't have. Two orange tigers producing a white fur tiger. Two parents with black eyes producing a son or daughter with blue eyes. Only possible if they have the heterozygous genotype, right? Or cheating, but the cheating is not an acceptable answer in biology, right? Okay, so only if you have the heterozygous genotype will that be possible. So this is your correct answer, and you have to write it in this method because this is how I taught it to you. None of you fools except Sanushi has written the correct way.
Me?
This like this.
Now I.
Now you
That is the whole thing.
Yeah. No, as long as it's like this, I can't see it from there. Bengal tigers, Bengal tigers have dark stripes on their fur. Keep it, I'll check it later. Suggest why each of the following is true for the pattern of dark stripes. It is not affected by whether the fur is orange or white. What's the reason? Now, why is the Bengal tiger? Now, Bengal tigers are orange with black stripes now. So why is having stripes? Why is it not affected by fur color?
It's coded for by different gene.
Excellent. The reason is whether the tiger is orange or white, he still has black stripes. It doesn't depend on the fur color. Having black stripes, meaning that does your hair two? Can you pay attention? What are you talking about? Huh? Pay attention, right? So if you have black eyes, okay, the question will say, in human beings, eye color is not dependent on hair color. That means if you have black eyes, you can have brown hair, blue hair, green hair, whatever the bloody hell hair you want to have. What is the reason for this? The reason for this is the gene for eye color is different from the gene for hair color. They are two separate genes. So whether your eyes are blue or red or not, that doesn't mean your hair also has to be the same color as your eyes. They're separate genes.
So then the fur only has these stripes, right?
Yeah.
The white fur. So then the part of the fur is the black stripe.
No, so the color and top of that there are stripes now. So it's saying here it is not affected by the fur. If the fur is orange or white, you still have black stripes. Why is that? What is the reason? Why is it not dependent on, why is black stripes not there for white but they are only for orange? There is nothing like that. It's independent, right? So the reason for this is it is controlled by different genes. It is unique to each individual.
Like some people wear black and some white.
So it is the pattern, right? The pattern of the stripes. Pattern. So some tigers can have stripes like that. Some tigers will have stripes like this. Some tigers will have stripes this way. Some will go this way. So it's independent, right? So remember that. So the pattern is coded for by a different gene. It is unique to each individual. The reason is why is the stripe pattern of stripes different from one individual to another? Because of the combination of alleles. The genotype. Yeah, because of sexual reproduction, because of mutations, the combination of alleles can be different. So the pattern can be different. That is the reason, right? No, not you will never find two, but one individual to another, if you look at it, it's different. Not that you can never have it again. Right? So this is because there are different alleles and as a result of mutations, the variety of alleles is increased. Huh? Huh?
There's a hashtag there.
Yeah, what meiosis results in different allele combinations. Quickly. Because they know there are fools who will write it for 6 lines. That's why. Quickly, guys. MEIOSIS.
No, I thought there was a script for the both after that.
So in this definition of
I did see that.
How did you see it? The statements E to K relate to the process of reproduction. Your favorite chapter. The most interested person. Only 6. Ayo, shit. Okay. Now, statements E to K relate to the process of reproduction. Table 5.1 shows a comparison of sexual and asexual reproduction. So, complete the table 5.1 by writing each letter in the correct box to match it to sexual reproduction only, asexual reproduction only, or both. The first letter has been written in the correct box. For you, use each letter once only. So, produces genetically identical offspring. Asexual only. H.
Oh, he.
Sorry, E. Sorry, E. Yeah, it's already there, no? Sorry. Produces more individuals of the same species. No, man. Both, sorry. Both produce. Asexual reproduction produces more individuals. Sexual reproduction also produces more individuals. More individuals is more members. Involves only one parent, asexual reproduction. Yes, they are usually clones from only one person. Involves only one parent, involves fusion of nuclear, sexual reproduction, not sexual reproduction. This is not a place for you to talk about your fantasies, right? Requires gametes, sexual, asexual, you don't have gametes. One cell, like in mitosis, divides to form another cell. forms a diploid zygote. Both. Zygote is produced. No. Zygote is only formed as a result of the fertilization. So if this said forms a diploid cell,
Should have been both.
it would have been both. Why? Because mitosis through cell division, it forms a diploid cell, meaning the same number of chromosomes in the parent are in the offspring. And sexual reproduction, when the two gametes fuse, it forms a diploid zygote.
A goes to
But
the-
this term zygote is only used for the cell that is produced after fertilization. So J is over here. Involves only cell division by mitosis. K, asexual only. That is your answer.
Allah.
In sexual, there is no mitosis.
These are both.
Until the zygote is formed, is sexual reproduction. Describe the role of section B. Right. So remember your old format. You can, when it comes to section B, you have to answer all questions. When it comes to section C, you're given a option. Either question 8 or 9. Anyway, we will do all since we are practicing. Describe the role of cilia in the trachea. What is the purpose of cilia? It is not to trap. Mucus traps. Cilia moves it away. Through its beating action, it moves the mucus. Right? It's nice. You know why? When I said beating, you looked at Abdullah only and you started laughing. Right? So now if you look at the lining, when you look at the lining of the respiratory tract from the trachea to the bronchi, to the bronchioles, right? The lining contains these hair-like structures called cilia that are constantly moving. What cilia does is, it takes the mucus and it sweeps it away from the lungs towards the throat, where you either spit it out or you swallow it, right? One of the two, right? So remember that. So cilia moves mucus containing bacteria, or you can say pathogens, whatever, and thus away from the lungs. This prevents infection. Why does it prevent infection? Because the mucus, now if this is the mucus ball in your bronchi, this mucus will contain bacteria trap. It will be dust. If the mucus is not moved away, this bacteria are present in the perfect environment. Why? Mucus is rich in nutrients, rich in proteins, rich in lipids, rich in carbohydrates. The air in your lungs is moist. The temperature is warm. There is enough of oxygen. This bacteria can easily multiply and cause severe infections. This is the reason why. This is the reason why sweeping the mucus away from the airways is important. Clear?
So what if they swallow them again? They are just swallowing the again.
What?
So if they don't speak it and then swallow the mucus,
It's destroyed. If okay, what happens? Why is swallowing mucus containing bacteria not harmful? Because of what? Huh? Because our stomach contains hydrochloric acid that kills the bacteria. Right? So swallowing the mucus, no harm, because the acid in the stomach can destroy the bacteria. The acid in the stomach has two roles. One, to provide an optimum pH of around 2 for pepsin. Number two, the purpose of the acid, the hydrochloric acid in the gastric juices, to ensure that the pathogens, it kills any pathogens that can enter. Figure 6.1 shows components of the human gaseous exchange surface and the associated blood vessels. State the characteristics. I have asked you this question for an exam. State the characteristics and describe the roles of each of the components shown in figure 6.1. You should make reference to name structures in your answer. So let us quickly, right? Let us quickly talk about this, right? This is what is the structure? The capillary. These are the red blood cells. This is the, what is this here? Yeah, the one cell thick wall of the capillary. This is also known as the capillary endothelium. The walls of the capillary is known as the capillary endothelium. Yeah. Huh? One cell thick. One cell thickness is one cell. You call it thin walls or one cell thick. No, can't. You want to talk to her? You want the phone? Why? No need. She'll take care of you. How many sisters do you have? I'm on the phone calling mother. Right? When you look at the, now this balloon-like structure is known as the alveolus. Alveolus is singular, alveoli is plural. Anyway, they'll be okay with it. You can see one cell thick wall of the alveoli. The alveoli is one cell thick. Alveolus is one cell thick. And this, what is this dotted line on the surface? It is a layer of moisture. Gaseous exchange surfaces need to have a layer of moisture. Why? Before the gas enters, before oxygen enters the blood, it is dissolved in this water and only enters. It enters as a solution, enters the blood as a solution. From there, it goes then into the red blood cell and binds to the hemoglobin, right? So this layer, if you look at the tube, where is the tube? Gaseous exchange. Here, guys, here, everything is here, right? Everything is in your tube. Large surface area to volume ratio, the alveoli provide. Why? In our lung, we have so many alveoli that if you were to open it up, you can fill one entire tennis court. Large surface area available for diffusion. One tennis court, if you open the alveoli in our lungs and put it flat, you can cover one entire tennis court. That is the, no, may all the alveoli mean tennis court. Why? They're small. Alveoli are thin and single layer of cells to minimize diffusion distance. Ventilation. Ventilation, that is breathing. Breathe, guys. Stop. Breathing in and out maintains a concentration gradient. Good blood supply maintains a concentration gradient for carbon dioxide to leave the blood and oxygen to enter the blood. A layer of moisture on the surface which helps diffusion as gases dissolve. This has all of this is in your tube, right? So let us go ahead. So this is a layer of moisture here. So let's go. The characteristics of the structures above enable efficient exchange, or you can say gaseous exchange of carbon dioxide plus oxygen through diffusion. So this is the purpose of having all of these characteristics, to ensure that carbon dioxide leaves the blood as quickly as possible and oxygen enters the blood as quickly as possible. Then you have to name the structures. You have to show them that you know the names, like the way we label. The alveolus contains a large surface area. You don't have to now repeatedly write alveoli contains large surface area for efficient gaseous exchange. Alveoli has one cell thick walls for gaseous exchange. We have already written that part at the top. The examiner knows now that since you have written the characteristics of the structures above enable efficient exchange of carbon dioxide and oxygen, you have written it. Examiner knows that whatever points you're going to write now is all because you're trying to prove that the characteristics for efficient gaseous exchange are present. So don't keep writing this increases gaseous exchange, this increases. You have written it once, the examiner understands, right? That's how you reduce your time. Alveolus contains a large surface area and a one cell thick wall that reduces diffusion distance, right? The one cell thick wall reduces diffusion distance, right? Enabling faster diffusion, right? So that reduces diffusion distance. Diffusion distance. Huh? Yeah, we'll talk. We are getting there, right? So next is it has a layer of moisture, allowing gases to diffuse, to dissolve before diffusion. Capillary has one cell thick walls. Once again, it reduces diffusion distance and connects to arteries and veins and ensures, and numerous capillaries. There's many, because there are many capillaries, there is a good blood supply. Ensure good blood supply and flow. Red blood cells, how do they allow for efficient gaseous exchange? Red blood cells have no nucleus. So why is this important to gaseous exchange? No nucleus means more space for hemoglobin. Means more hemoglobin can be present to carry oxygen. Also, the other adaptation is they are biconcave in shape. Yeah, which the biconcave shape allows them to increase the surface area for diffusion. And another point, so that you don't have to write biconcave, you're just supposed to write the characteristics now. And then also you can write another characteristic, they live for 90 to 120 days. They're there for a long time. So they can carry out their job for, yeah. Can be present to carry oxygen. They are biconcave in shape. They live for 90 to 120 days. Which, Huda, what were you asking? Everything that is written is an answer. In what B, Huda, how are the marks allocated in B, meaning which? Anyway, it's okay. Question for, I'll get back to you. You guys write this answer, will you? What? Yeah, why are you so interested about what I did when I was young? Huh? Now you see how your past papers, when you now, for example, day before the exam, if you read this past paper, you're also revising your theory. What is the theory? You're revising the characteristics of gaseous exchange surfaces. You're revising so many things. And you're also revising, when you read this past paper answers, how to write the answer. What are the important points? So if you get another question like this, so usually questions are repeated, not the exact same thing, but they will give you a different situation, but ask the same question. You can just simply remember this answer and write it down.
They
Yeah.
live for the last before this participant.
They live for 190 to 120 days.
So the 7 marks question, they wanted the whole thing, right?
No, the whole thing.
This is a.
7 points from this. So basically you get 1 mark for saying that these characteristics allow efficient gaseous exchange of carbon dioxide and water. Alveolus contains a large surface area, another second mark. 1 cell thick wall, third mark. Large layer of moisture, fourth mark. Right? Capillary, 1 cell thick wall, fifth mark. Connects to arteries and veins, 6 mark. Allows good blood supply and 7 marks. Red blood cells have no nucleus, 8 marks. So these are you only have to get 7. What I always tell my students is, if a question is asking for 3 marks, you give 3 plus 1 extra point, 4 points, just to be on the safe side. So question asks for is a 3 mark question, you have to write 4 points, right? 4 points, the 4th point, basically each point is about 2 or 3 words long, right? If you write it summarized in a summarized way. So if a question is asking for 7 points, you give 8. Because why? Sometimes they will consider 2 points as 1 mark. So just so that you don't lose, you write that extra point just in case. Of course, for a 1 mark answer, don't write 3 points. That's ridiculous, right? 8 marks, 9 marks, they are biconcave. They live for 9 to 120 days. Yeah, I'm writing all the possible answers. So you write, if you're writing the answer in the exam, you write it till 7 marks. But I am writing all of this so that you know what are the other answers you can write. A full review. Huh? Okay, next. Figure 7. Figures. Shafiq. Hey, darlings. 7.1 shows bacteria growing on a surface of a dish containing nutrient jelly. Paper dish such as M and N was soaked in solutions of different antibiotics and placed on top of the growing bacteria. A clear area on the jelly indicates that bacteria in that area have been killed. Use the information above and your knowledge of the process of natural selection to describe and explain the difference in appearance of the jelly surrounding disc M and disc N. How are you going to write this answer? Lovely question. Now, first of all, what does the clear area indicate? No bacterial growth. Why is there no bacterial growth? No, my question is, what does this clear area indicate? No bacterial growth, right? Okay. Why is the bacteria not growing? So what they are doing is they are taking this basically paper and they are soaking it in different antibiotics. They have antibiotic A, B, C, D, E, F. And they are putting it here like this. A, B, C, D, E, F. Now, after soaking this paper disc in an antibiotic and putting it on a dish where the bacteria is growing, if the antibiotic is effective, then you will have a clear area. Clear area means bacteria is not growing. Here, this area here, this is all shaded. That means bacteria is growing, right? Now, based on that, what are the least effective antibiotics? These two now. Yeah. What is the most effective antibiotic? Most likely this. Yeah. Why is this? Because the area, the clear area is more, meaning that more bacteria have been killed and destroyed. Right? So, similarly, if you compare M and if you compare N, which one is antibiotic resistant? Which one is not antibiotic resistant? N is antibiotic. What is antibiotic resistance? Basically, some bacteria have alleles. They have mutations that give them the power to withstand the effects of an antibiotic, meaning that because of that mutated gene, the antibiotic cannot kill the bacteria. Bacteria is protected. So even if, for example, if you get an infection from a bacteria that has antibiotic resistance, then if you take, let's say, bacteria is antibiotic, the antibiotic is resistant to antibiotic. The bacteria is resistant to antibiotic X. If you take antibiotic X for an infection caused by this bacteria, what's going to happen is you will not kill the bacteria will continue to survive. What's going on? Constantly, I see you guys talking, and you too. Don't disturb, please, right? In this class is going on, pay attention. So antibiotic X is not who is smacking the pen on the table. Are you done or not? Are you done? Yes. Keep that pouch inside under the desk. Okay. So antibiotic X, you have a bacteria, and this bacteria is resistant to antibiotic X. If you have an infection caused by antibiotic X, right? No, sorry. If you have an infection caused by this bacteria, if you use this antibiotic against that infection, will the bacteria die? It won't die. Why? Because the bacteria already has the ability to resist that antibiotic. That is what's happening here. The antibiotic which is present on this disc has no effect on killing bacteria. That's why the bacteria is just growing around it. The antibiotic present here on disc M has killed bacteria. That's why there is a clear space. Understood? So use the information above and your knowledge of the process of natural selection to describe and explain the differences in appearance of the jelly surrounding disc M and N. So basically, you have to give the idea that disc M bacteria are not resistant to the antibiotic in disc M, which is why they are killed. Antibiotic, the bacteria is resistant to the antibiotic in disc N, which is why they are not killed. That is why there is no clear space, right? The reason for this is, how did I teach you when I taught you about antibiotic resistance? What did I tell you? You have bacteria, several bacteria. One bacteria develops a mutation. This mutation, meaning there is a change in its gene, and that changed gene produces a protein that can withstand an antibiotic, resist an antibiotic, prevent an antibiotic from affecting it. What happens is, if you use that antibiotic, this bugger will die. He doesn't have this antibiotic resistant gene. This one will die. He doesn't have the antibiotic resistant gene. This one will survive. He survives. Why? Because this mutated gene is giving him a survival advantage. He survives, and what happens? He produces through binary fission, which is a type of mitosis. He produces offspring, and these offspring also have this mutation that allows antibiotic resistance. So because of this mutation X, this bacteria survives, reproduces, passes on the mutation to its offspring. Now in your body, because what did I tell you? Some antibiotic practices are not good. We are asked to take antibiotics for 5 days. We take it for 2 or 3 days. We are told to take, now that's how our mothers are, no? Honey, I'm sick, go to a doctor. Doctor gives for 5 days. After 2 days, no sore throat, stop taking antibiotic. Next time, more severe infection, antibiotic is not effective. Why? All the useless buggers have been killed. And because you have not taken the antibiotic properly, only the one that has resistance survives. And in the end, when you get the infection again, it is by you're being infected by these bacteria that have this mutation, antibiotic resistant mutation. Why? Because you didn't take the dose properly. You have killed the bacteria that are weak. You have allowed the bacteria that has the mutation to survive. He survives, he reproduces and passes on the mutation to others. And now you have a full army of bacteria that can resist the antibiotic you're taking. So we studied that there are 2 practices that are increasing antibiotic resistance. Patients are not taking their medication properly, the antibiotics properly. They're not taking the correct dose. Doctor says 500 milligrams 3 times a day. They take 200 milligrams once a day. Break the tablet in half and they take.
Then why don't you- They're getting the same.
Because how can you explain to our patients, man? They don't know anything, what to do. You try to explain antibiotic resistance to the patients, we have to spend 1 whole day on a patient. You buggers itself can't understand. How do you expect others to understand, man? Right. So use the info. So what you have to say is M has a clear area and N has no clear area. Bacteria have been killed around M but not N. Right. So you can say that for disk M, contain an antibiotic that was able to kill the bacteria as the bacteria did not have any genes for resistance. To antibiotic, sorry, not to antibiotic resistance, right? Disc N contained an antibiotic that the bacteria was resistant to. The bacteria may have got a mutation in a gene which allowed, which gave them a survival advantage enabling them to reproduce and pass on the allele to their offspring, which spreads the resistance. Sorry? Next. Because the patients have driven us mad. Now we don't have control of the way we write. Describe how the process of artificial selection differs from that of natural selection. Include reference to the production of one named economically important plant or animal in your answer. So a plant, basically, they're asking you to describe artificial selection from an example of a plant or animal that you know of. So you can talk about mangoes, right? One type of mango is large and sour, one is small and sweet. So you want to produce a tree that mix the best of both features. So you have one mango, large but it is sour. The other mango is small, but it is sweet. You want to produce large and sweet mangoes. What you do is you keep crossing, right? You take two individuals. The reason mangoes is, now why are mangoes economically important? Because as a farmer, if you're having a mango farm and I'm a customer, will I like to buy small and sour mangoes or small and sweet mangoes, or will I like to buy this one, the large and sweet mangoes? So I will love to buy large and sweet mangoes, but these trees are very few. So as a result, if I am a farmer, if I want to make the highest amount of profit, because you guys want to buy large and sweet mangoes, if I have very few of them, but many of the useless ones you don't like, I will lose my business, right? All my hard work, all the money I invested to farm and grow these trees, all will go to waste because nobody will buy. The fruits will go bad and I will lose. So as a farmer, in order to make sure that I am giving you the best, the product that you want, I will take trees, I will cross-pollinate them and repeat it over many generations until I finally get a tree that produces, that has both the characteristics, produces large mangoes and sweet mangoes. So this answer that I'm going to write, you can use for any artificial selection question. So keep it in mind. So it's not only for trees, you can take two different types of goats or sheep or whatever, and you can use it, right? So a mango, wait, not mango tree. Mango trees can produce either large but sour mangoes, or small sweet ones. In order to produce large sweet mangoes, which improves sales, farmers cross. So why is it important to say the farmer or a man or a woman or whatever is doing this? Because no, man, because there are two types of selection: natural selection and artificial selection. Artificial selection where human beings are crossing two individuals, two plants or two animals or whatever, in order to get some characteristics that are going to make us profit, to favor us, to help us. Natural selection, there are no humans involved. Naturally, it is happening because of how nature is, right? So you have to mention farm or human to show that this is artificial selection you're talking about, because humans are putting their hand and changing things, which improves sales. Farmers cross breed the two mango plants over mango plants and their offspring. Over several generations till they produce a tree that can produce large and sweet mangoes. And this is the tree that they will keep. This tree they will keep in the farm because the other trees are not favored. Okay. Now, wait a sec. Actually, June 2017. Paper 2. Now, for the next class, guys, that paper has been uploaded onto the group. We will be very high dose of antibiotics can cause side effects actually to your body. So May, June 2017, paper 2-2. So we have almost done with paper 2-1. We'll complete 2-1 in the next class. Get ready with paper 2-2 and come.
Is there possibility, for example, antibiotic can start killing the ones like there are resistant, and like small amount is resistant, like mutation, and it starts reproducing and it starts reproducing like it's like something you feel it.
Repeat it.
For example, in was before like this, you kill all the non-resistant ones, and one mutation became resistant. It started rapidly reproducing. The resistant one is like growing in the area, and so it's like the bacteria is resistant.
The one more.
There are possibility.
I don't understand this question. What's the bacteria in this?
The bacteria is this white
Shaded
part.
part. Shaded, shaded.
White area, yeah.
Very good. This.
Shaded.
No, all of this entire thing is the bacteria. This white dot is a paper that is soaked in the antibiotic. Now bacteria is growing in this, they are putting the white dot and they see is the growth effective or not.
This is not effective. This is effective.
Yes.
So,
Okay. I will have to give you. I'll forward the PDF. 4B. 4B, the marks are allocated.
No.
Let me see, I can't even remember. So 1 mark to show heterozygous, right? 2 heterozygous individuals. 1 mark to show the formation of the homozygous, sorry, one mark to show the gametes, show the gametes. Second mark for the formation, the result in the formation of a homozygous recessive, which is the white phenotype. The third mark is to show the labels on the genetic. So 1, 2, 3, right? Those three. Right? Understood? Okay, see you next week. Take care. Bye bye.